Thank you for inviting me to speak to you today about fish contaminants in Lake Michigan. For those of you who don't know me, I coordinate the fish contaminant monitoring and advisory program for the Wisconsin DNR, and I work with human health epidemiologists and human health toxicologists from the Great Lakes states and other states and our own Wisconsin Division of Health. Also fisheries biologists who do a lot of the collections. And it includes the Great Lakes states, Great Lakes Fish Consumption Advisory Consortium exists to develop protocols on how contaminant information is put into fish consumption advisories. So today I'm going to focus on the important pollutants that bioaccumulate in fish, touch on a little bit of what we know about the factors that affect accumulation in fish and the temporal trends of the contaminants. And some of this information I'd like to acknowledge is from others, including Beth Murphy, who coordinates the US EPA Great Lakes National Program Office, Great Lakes Contaminant Monitoring Program, and others whose information I'm using are listed on the slides. So most of you probably know that the contaminant, bioaccumulating contaminant of greatest concern in Lake Michigan fish are polychlorinated biphenyls or PCBs. And this chart here is from a recent publication by McGoldrick and Murphy, where they summarized the last 5 years of their contaminant monitoring. And this is the program is comprised of whole fish lake trout. And you can see that on a mass basis, PCBs are number one, with the average concentration in the whole fish just less than 1 part per million. And then secondly, with the second mass is the chlorinated pesticides treated as a whole group. And then third is mercury, and lastly, recently EPA, Great Lakes National Program Office, added some new emerging contaminants to the list of analytes. So I'm not going to really discuss those today, but just be aware that we're gathering more information about those contaminants. Today I'll mostly focus on PCBs and mercury. Probably most of you know that PCBs are lipophilic. They're a man-made group of chemicals, very resistant to degradation and highly bioaccumulating. They since their time of production and use until restrictions were put in place on their production and use, the PCBs that were discharged sorbed to sediments and moved throughout the environment, accumulated in fish and also in fish-eating wildlife and fish-eating humans. And then in comparison, mercury is an element of the earth. It exists in a variety of different forms and travels throughout the atmosphere and with weather systems. It changes into different forms, and it's the methylated form that accumulates to the highest concentrations in fish, and it is also the form that is the most toxic to fish-eating wildlife and humans. And in order for the mercury to become methylated, it requires the presence of anaerobic methylating bacteria. So over time and through a lot of different studies, we've learned about the different factors that affect the concentrations of mercury and PCBs in fish. And we know that mercury concentrations and PCB concentrations that are achieved in fish vary between the species, between water bodies. Of course, for lipid, a lipid content only affects PCB concentrations. And then some of these other things we're just beginning to learn about how PCB and mercury concentrations vary with seasons and between genders and with their diet and different sorts of factors like energetics. Wisconsin DNR has tested fish since the 1970s, and most of our testing now is comprised of fillets, and that is because we use this information to update our fish consumption advice. And this chart shows the 5th to 95th percentile of the PCB concentrations in primary species from our database for the last 10 years. And you can see that lake trout achieve the highest PCB concentration and also have the greatest range of concentrations. And they also have the highest lipid content for those species presented. And looking further at lake trout, this slide shows PCB versus length and uses data from the other Lake Michigan states. And you can obviously see the increase in concentration with length. But what you can't see is, until I put up this other slide, is that it seems like some of the concentrations vary by the source of the data, perhaps representing where the fish were collected, or it could also be due to other factors other differences in our monitoring protocols. For instance, Illinois tends to collect their lake trout in the spring and they composite their fish. So there'd be like 5 fish in a sample. Michigan's data is all from the fall of 2000, and I'm thinking most of their fish are from the northern part of Lake Michigan, if I remember correctly. And Wisconsin tends to collect most of the lake trout from summer, and we test individual fish. So there's a question of, where does this variability come from? Is it just from length, gender, and or is it from location and other sorts of factors that maybe we could suss out a little bit with further work? And then moving on to trends, this again shows fillet concentrations of PCBs over time in, it's basically a big mashup of all our fillet data, our Wisconsin DNR data, and you can see that PCB concentrations have dropped over time. You can also see that the fillet or the lipid content has also decreased over time, at least for the species that are represented in each one of these time periods. But it does show you that people eat these fish, so people who are eating fish are taking in less PCBs than they used to. So that's a good thing. Going back to the Great Lakes National Program Office, as I said before, they've conducted whole fish lake trout monitoring in each of the Great Lakes since about the 90s or maybe the 70s, but you can see that Lake Michigan and Lake Ontario have the highest concentration of PCBs in the whole fish lake trout. Lake Erie is where they collect walleye, shown in blue. So you can see these graphs also suggest a decreasing concentration in PCB, decreasing trends in PCB concentrations over time. And this presents the lake trout whole fish for Lake Michigan in particular. This is EPA's data, and you can see the decreasing trends. Now, the point I wanted to make about this slide is that EPA's program was originally designed to be standardized in some fashion. So their collections are all from fall. They composite typically five fish into one sample and test that sample. They also have a size range, so they target 600 to 700 millimeter fish for their collections. However, what they've recently realized that standardization by size doesn't necessarily mean that you're going to get standardized data or consistent data over time. And what they've recently found is that the dots represent the actual fish that were collected, and that over time the fish that were actually collected drifted up in size and in age at a different rate, and at times were outside of their targets, which are shown, the targets are shown in the shaded boxes. So that leaves the question of what sort of trends do you want to include in the State of the Lake report? And what might be useful is to look at PCB trends in a variety of media or in a variety of species. And recently, Chang summarized some of these studies, and this chart shows the estimated annual decrease in PCB concentration in a variety of different media, lake trout, and these are for each of the different Great Lakes. So this information could be useful in, verifying the trend that you're presenting for one species or another, and it might also be more important in the future as hopefully PCB concentrations change, that if we have information from a variety of media or a variety of species, it may be useful in figuring out the mechanisms that are driving the changes in PCBs, whether it be sediment remediation or perhaps changes in diet or any kinds of fish community changes that might affect PCB concentrations in fish over time as Lake Michigan changes. So recently, with assistance from our Wisconsin DNR statistician, we were able to come up with an estimate of the temporal trends of PCB concentrations in Wisconsin DNR Chinook and salmon fillets. And what we found is that the best fitting model, and a number of models were evaluated, but the best fitting model included factors that accounted for the two different time periods, and also the season that the fish were collected, the percent lipid in the fillet, and then the length of the fish. So you can see each dot represents one of the records of the fish included in the exercise, and then the two lines represent the time trends for the fall and then summer fish. So we think this sort of exercise is needed to come up with a good estimate of changes in PCB concentration in whatever species you want to look at. In some cases, some of these factors may not be significant, but for Chinook, this is what was important. We were not able to include gender or age, primarily because while we had some information on gender and age, it wasn't sufficient to add anything to these models. So then, moving on to Mercury, this is the same sort of mashup showing for Lake Michigan Wisconsin DNR fillet data for mercury. The reason I'm presenting this is compared to lake trout with the high fat and the high PCBs, here we find that yellow perch have the highest concentration and greatest range of mercury concentrations. This is a little limited in our sample size, but I think it bears some follow-up. Most of these fish were biased towards collections off of Milwaukee, so one could wonder if it's just because these yellow perch were able to attain old age, or is it because there's some kind of methylation present near Milwaukee. So this is kind of interesting, and it also shows that while PCBs are the main pollutant of concern for most species in Lake Michigan, that's not always the case. In some situations, mercury is a greater concern. And you can see this is both mercury and PCBs in the yellow perch from Lake Michigan, using data from Wisconsin and Illinois and Indiana just from 10 years. And you can see that mercury is a greater concern compared versus PCBs in the yellow perch. So unfortunately for mercury, we're not really able to say too much about the temporal trends of mercury in fish over time. Primarily, one reason is that prior to about 2000, very little mercury testing of fish in fish was conducted because the primary concern was PCBs. However, other studies have been done, and recently Great Lakes region-wide effort was conducted to gather all mercury data for walleye and largemouth bass from all the different areas, and similar exercise to what was done with the PCB, where we looked at a number of different models and came up with the best model and estimate of temporal trends. However, for this estimate, Lake Michigan was not represented, so maybe in the future we'll be able to look at changes in mercury concentrations in fish from Lake Michigan. So besides PCBs and mercury, I just wanted to mention that there are other bioaccumulating contaminants in fish. Most of these most of the legacy contaminants have decreased. And by legacy, I mean the banned pesticides, dioxin, that's probably about it. But it's still, it's good news, so that's something to report on. And finally, in preparation for this presentation, I was wondering what sort of information the State of the Lake report included in the past, what kind of information you were looking for. So besides presenting information on trends in others, in more than just one species, in the State of the Lake 2000 report, there was an estimate of the amount of PCBs removed from the Lake Michigan basin. I don't know how difficult that would be to obtain, but that could be something that you would want to include in the report. And I also wanted to just mention that I focused only on the bioaccumulating chemicals. There's a whole, we're learning a lot more about a whole variety of substances that are in the water that don't necessarily bioaccumulate in fish. And the water is the fish's habitat and may be affecting fish health. And these include things like pharmaceuticals, personal care products, microplastics, new kinds of chemicals that are not bioaccumulating. And then Also, there are some recent publications on some human biomonitoring that is conducted of legacy chemicals and also emerging chemicals like the perfluorinated and the polybrominated diphenyl ethers. So, I mean, these studies include humans that ate Lake Michigan fish, so there could be some information there that you might want to use for the State of the Lake report. And then lastly, I just wanted to mention that I do work with the Great Lakes Fish Consumption Advisory Consortium, and this consortium is comprised of health epidemiologists or toxicologists and a few environmental monitoring people like me, and we develop protocols for how we issue advice, and we also examine contaminant data. One thing that I would like to see more of is information from the fisheries findings incorporated, especially like where fish move around and things like that. I think that would be beneficial to some of the decisions that we make with regard to contaminants in fish and fish consumption advisories. So, in summary, PCBs are the main pollutant that are the main bioaccumulating contaminant of concern in Lake Michigan fish. Concentrations have declined, and what we're uncertain of is the rate of future decline, because given Lake Michigan changing environment, it's difficult to make those sorts of predictions. Although at one time there was a prediction about lake trout. I'm not sure how valid that is anymore. So with that, I'd be happy to answer any questions.